For those of you that are watching this recording back, I did start a little bit late, essentially. We're into chapter 3 on cell biology and just quickly address the size of cells. And now we're going into the major organelles. So since this is a human anatomy and physiology class, we are focusing specifically on animal cells, since that is what our bodies are made out of. So we're not going to be addressing cell walls or chloroplasts or really even vacuoles, since they're rare in animal cells. But all of the organelles you see here are in all of your cells, essentially, or majority of your cells. There are some exceptions. Okay. So general cell structure here. Cells are three-dimensional. So even though a lot of the images we look at are 2D to give us a flat image, you should be thinking about your cells as three-dimensional things. Right? They exist in our universe where all things either take up space or are empty space. Our cells are completely surrounded by what is called the plasma membrane or the cellular membrane. And then that is what determines what is inside that cell versus what is part of the extracellular environment, so what is outside of that cell. Inside of the cell, a majority of the space is taken up by a aqueous liquid, so mostly water-based liquid called cytosol, cytoplasm. The cytosol, no, I'm switching that. Let me back up. The cytoplasm is the term used to describe all of the interior of the cell, which includes the cytosol, which is that aqueous liquid, and then the organelles. So those two components together make up our cytoplasm, which is what fills the space in our cell. So cytosol is the liquid outside of all of our organelles, and then the rest of the space is taken up by organelles. You can see some of the major ones here, but we have slides talking about all of them in particular. We're going to come back to centrosomes and centrioles. It's just kind of breaking apart that, that larger image. So throughout our cells, like I said, we have the plasma membrane completely surrounding it. This is mainly made up of lipids, those phospholipids, and a bilayer formation with proteins and carbohydrates attached to it. But we also have extensions of those proteins that go throughout the cell, giving our cell structure, sometimes extending outside of the cell to communicate with other cells or connect with other cells. And those protein fibers are called our cytoskeleton. So cyto referring to cell. So this is the skeleton of the cell. We don't have bones inside of our cells, but we do have these protein fibers that help give some structure and form to the cell itself. Okay, we're going to talk more about cilia later. We'll talk more about all of these essentially, except ribosomes. So I guess we can, I have a slide on ribosomes. Ribosomes are a very plentiful organelle in all cells. Ribosomes are a required organelle for a living thing to function. Ribosomes are these teeny tiny protein-based structures you see floating around here. And these are the sites of translation, essentially, where our genetic information is going to be turned into an actual protein. So ribosomes are super, super critical for actually carrying out the functions that are coded for in our genes, in our DNA. That DNA needs to be transcribed and then translated at the ribosome in order for it to make an actual protein. Okay. The plasma membrane of cells is probably one of the most complex structures within the cell to talk about and think about. And it has a really big role to play. Okay? It is essentially responsible for maintaining homeostasis within that cell. So ensuring that the conditions within the cell are within the correct range for that cell to continue functioning. And it plays that role by being somewhat selective on what actually gets to cross this membrane. So what gets to enter the cell as well as what gets to leave. So the major structure of our plasma membrane here are these phospholipids, or this phospholipid bilayer. And we talked about this a bit in the last chapter, right? But these phospholipids have a glycerol head with two fatty acid tails, and then they are attached to a phosphate group at the top here. And that phosphate group gives a slight charge to the glycerol heads, which allows them to interact with water. Right? Typically, lipids are non-polar and thus do not interact with water, or fatty acid tails are always that way. And so that phosphate group giving a bit of a charge, giving some polarity to the heads, allows them to interact with water. And thus we get this formation of phospholipids where the hydrophobic tails, which cannot interact with water, point inwards, preventing the free movement of many molecules across it. And then we have our hydrophilic heads pointing outwards in both directions. And since they can interact with water, they're not just simply being pushed away like the tails would be. And if we didn't have these heads, the entire membrane structure would fall apart because water would be pushing away the tails, or tails would be pushing away the water, and the whole membrane dissipates. And so in this way, we have heads pointing towards the extracellular environment, which is almost always going to be some aqueous fluid, some water-based fluid, and then heads pointing towards the intracellular environment, so inside the cell, which again is most of the space inside the cell is full or is filled with cytosol, which is another water-based aqueous solution. All right. So this formation, like I said, prevents the free movement of lots of different molecules in and out of the cell. And I'll come back to that later, but essentially, if it is carrying a charge or it is a large molecule, it is not going to be able to pass through this membrane without the help of these membrane proteins. And so we have lots of different types of membrane proteins that get embedded into our membranes. Some of them create channels, like a tunnel, that are just always open for particular molecules to travel through, either into or out of the cell. Some of the proteins are gated, meaning they need to be activated in order to open. Some of them completely change shape to move a molecule from one side to the other. And then some of them are there to hold other molecules, such as these glycoproteins that we have indicated here. Glycoproteins are proteins that are embedded into the membrane that have carbohydrates sticking off of them. And those carbohydrates that are in the extracellular environment are what are used by neighboring cells to recognize each other. Right? These can interact with other proteins or make bonds with other glycoproteins, so the other carbohydrates, and that is used to transmit signals and to help lock each other in place. So neighboring cells can hold together and stay together. And then the last thing to mention here is cholesterol. So cholesterol is another type of lipid, but cholesterol stays relatively solid at higher temperatures compared to the phospholipids that are mainly making up our membrane here. Phospholipids tend to be liquid at higher temperatures. So like our average body temperature, which is 98.6 degrees Fahrenheit, at that temperature, phospholipids are liquid. And so these more structurally stable cholesterol molecules can be added to or moved within the membrane to provide more stability in certain areas. All right. So the rest of this slide deck is going to get much more into the specifics of certain functions and certain structures. And so we'll start with the cytoskeleton, which I pointed out back here, right, is that system of protein fibers that are used throughout the cell to give it structure and shape. But they're also used for lots of processes like when our cells replicate, and they're used as like tracks for molecules to move throughout the cell as well. We have specialized proteins like kinesin, which essentially bond with, break bonds, and then bond with again to produce this kind of like walking motion. And there's a video that I've provided on Canvas that shows that at a molecular level, that kinesin protein just kind of waddling across a microtubule, which is kind of cool. And so our cytoskeleton, again, made up of protein fibers, and we have a few different types of protein fibers that are all going to be involved here. But each of them has some specific functions or structures that they play a role in. So the smallest protein fiber within our cells are the actin filaments. So actin is the type of protein, and then actin bonded with other actin make these long filaments. These are quite flexible and elastic because of their kind of spiral formation that they make, but these are the smallest version here. Our largest protein filament in our cells are microtubules. So just like the name suggests, they end up making a tube. They end up making these hollow cylinders of protein. And they have a quite specific structure because we have two different types of tubulin that are involved in making these microtubules. So you can see the different shades of green in this microtubule structure. We have a dark green and then a light green. Those are the two different types of tubulin. And that provides that ability for that actin protein that I was talking about that kind of waddles. One of its legs will only bond with the dark green tubulin, and one of its legs will only bond with the light green. So they kind of get to do this swapping, waddling, and move across these microtubules as if they were some track, like a railroad track. Microtubules are also often used for structures that are going to be quickly made and then broken apart, such as during cell replication, these spindle fibers that spread out and attach to sister chromatids and then are supposed to pull those sister chromatids to the two different sides of the cell. Those are microtubules. And so these tubulin dimers can be added really quickly to build those fibers out, and then they can be removed relatively quickly as well to help shorten those fibers and bring the two sister chromatids apart. And then lastly, we have our intermediate filaments. These are the most structurally stable of our protein filaments here. These are what make up a lot of the larger protein filaments like collagen, which is a major part of like our bone. And so again, smallest are actin, they're elastic, stretchy. Largest are these microtubules. They make hollow cylinders. They're used in processes that need to extend and then retract quickly. They're also used as a track for other proteins to move along. And then these intermediate filaments are the middle intermediate size, which are much more rope-like and used for some of the larger protein structures like collagen. And, so here's some more examples of where you can find these types of filaments. So intermediate filaments make up those support structures of the cell. So like keratin, if you've heard of keratin, keratin is an intermediate filament, a type of protein that makes up our nails and hair. Microfilaments, again, the smallest here. These are very flexible and they are used to help support the cell and are along the outer portions of our cytoskeleton, making up the terminal web for some other structures like our microvilli. But again, giving that elasticity and flexibility. And then lastly, our microtubules. These are what produce sort of the actual skeleton part of the cytoskeleton, stretching throughout the cell to give that cell shape, and again, act as tracks for movements in a lot of ways. So here's a look at some stained proteins within an actual animal cell, looking at it under a microscope or a micrograph here. So you can see the extensiveness of the protein network that is found within the cell. Okay. And so the cytoskeleton does not always stay within the cell itself, or at least can also play a role in extending the plasma membrane, kind of pushing the plasma membrane into these different shapes. And so we have two different versions of this membrane extension that come from our cytoskeleton in animal cells. So there's another version here that we're not going to talk about in much detail, which is the flagella. The only human cell that uses flagella is the sperm cell produced by biological males. And so for most of the cells we're looking at, or most of the structures that are going to be prevalent and play a role in the processes that we consider in this course, are only going to be the cilia and microvilli. So cilia and microvilli are very similar in that they are both these short extensions, almost hair-like extensions of the plasma membrane. So we have those two, those protein fibers pushing the membrane out this way, but they play different roles. So cilia do a lot more movements, and they kind of go in this flowing back and forth, what is called a power stroke in these cilia. And in like bacterial cells, movement of cilia in that way helps them move through their environment. But in our cells, these cilia are mainly there to move molecules across the membrane to help push them in some direction across the cell surface. Microvilli are very similar in that they are still these short extensions, and there's many of them, almost hair-like, outside of the cell. But the purpose of a microvilli is not to move at all, but rather to just increase the surface area of our membrane. So instead of this membrane being flat, all these extensions are increasing the amount of membrane that is interacting with the environment in this space. And so in areas in our body where we need to have a lot of movement of molecules into the cell, such as in our small intestine and large intestine, right? Most of the absorption of the molecules from our food is happening in the small intestine. And so the cells that line the epithelia pointing into our small intestine have a lot of these microvilli, and that gives a lot of room for molecules to interact with and cross the plasma membrane, which is, of course, what we want all of our food to do. Once we've digested it and broken it down, we want to get it into these cells and then into our circulatory system. Another distinct difference here, the moving version, these cilia are made up of microtubules, and then they're not necessarily stiff, but they aren't actively moving themselves. Those are made up of those actin microfilaments. Okay. So here's a look at another microscope image. In this case, we're looking at microvilli sticking out. So again, we have these microfilament actin protein fibers sticking through the plasma membrane, extending that plasma membrane. And something that's shown well here is what is called the terminal web. And so the terminal web is that outer ring that we saw here, these microfilaments in green that are all along the inside barrier of the plasma membrane. Those act as a root system for all these other protein structures that are extending outwards. Right? So the microvilli, they're the filaments that make these microvilli, are anchored by this terminal web. There you go. Terminal web is used to anchor microvilli. And you can see that a bit better here. It also anchors other parts of the cytoskeleton that extend into the cell itself. Oh gosh. I don't know if this picture has always been this fuzzy, but it certainly looks fuzzy today. We have 4 different types or classes of intermediate filaments. We're not going to spend a whole lot of time necessarily talking about them in depth, but you'll see them come up in a few different ways. So those keratin filaments I was talking about, which again are what make our nails and hair, are much more common in our epithelial cells. These are typically what is lining the outer portions of a tissue. Vimentin or vimentin-related filaments are much more common in connective tissues, muscle cells and glial cells. And then we have some intermediate filaments that act as neurofilaments in our nerve cells. And then lastly, we have nuclear lamins present in all animal cells, which is fibers that help make the membrane structure of the nucleus. So all of the organelles in our cells have their own phospholipid bilayer, the same way that our plasma membrane surrounds the entire cell. Each organelle has phospholipids that surround that organelle. But the membrane of the nucleus itself is slightly different. It has its own particular structures, such as these nuclear intermediate filaments on that membrane. Okay, so just another microscope. Okay, some more examples of key microtubules. So you're going to see this show up again later when we talk about cell replication. So a particular type of microtubule called centrioles. These centrioles always end up in this specific formation of 9 microtubule triplets. So we have 3 microtubules here, or microtubule polymers here, making those hollow tubes. And then we have 9 of those triplets that make this circular structure here. Essentially, these end up being the anchors that will extend those spindle fibers to attach with sister chromatids during replication. And then they will be the anchor where those microtubules are pulled back to separate sister chromatids so that we get 2 new cells with all the copies of DNA that were there originally. This is just a further look at cilia. You're not going to be necessarily tested on an exam about these specifics of these fibers, but it is in the text, so it's showing it here. Essentially, cilia have a very similar basal body with those microtubule triplets. I already talked about the power stroke. Again, this is just exemplifying that cilia move back and forth. And so they are really useful for moving molecules across the cell membrane, not moving them into or out of the cell, but moving them across the outer portion of the cell membrane. So here's that video I was talking about. So what's shown in this image, which again is part of the animation of the video, this is that kinesin transport protein that I was mentioning. It has two little feet here that will alternate breaking and recreating their bonds with the next tubulin dimer. So moving from one tubulin to the next one, doing a kind of waddle. And they can carry really large packages through the cell or through a space in this way. And so in this case, in this video, it's transporting this white blood cell or leukocyte, which is very, very large across this microtubule tract. It's kind of cool. Just something of note here, in one of my very first biology classes when I was still a psychology major, just seeing the action or seeing this kinesin protein move was one of the first things that I got like really excited and passionate about, that made me like want to go home and dive deeper into this stuff. So I highly recommend you check out the video. I just think it's cool to see human-like things showing up on a microscopic level in our cells. Okay. Or I guess before I move on, are there any questions or concerns or points of clarification on the cytoskeleton? And just to get some quick feedback on how I'm doing with this. Am I going too fast? Is this an okay pace? Would anyone like slightly slower, slightly faster? Any kind of feedback you guys can provide.
Well, I guess that'll depend on how much is left.
Well, we have 99 slides in this slide deck to get through today and tomorrow, and we are on slide 24. So we're about halfway through with what we need to get done today. Meaning I have more than enough time. So could certainly slow down if there's anything you guys want me to expand on.
I guess you could just go finish up at the pace you're at, and then I don't know. It's maybe not, but I was thinking that maybe you could then re-mention the most important things to reinforce them twice.
Yeah, absolutely. And that is my general plan. Anytime that we work through content faster than we need to get through the content, that usually means the next class I'm going to have some sort of activity designed for you guys to help dive into the deeper concepts and then allow me opportunities to expand on those ideas. So that is going to be the plan, right? It seems like we're getting through this relatively fast, which is great. And that means next class I can have some activity for us to work on to help with develop that understanding.
Yeah, I was gonna say you're fine. Like, I like that you record your lectures, so then like I can listen again. I mean, and that's good enough for me. And then honestly, like I rewatched the chemistry one, and a lot of those answers were on the quiz. So like, yeah, you're fine.
Awesome. And the cool thing with the recorded lectures, depending on how much time you spent on YouTube, you may or may not be aware, but you have control over the playback speed on YouTube. So like I was talking really fast in the chemistry video to get through that content. You can slow that down to 0.8 or 0.75 as long as that doesn't make my voice sound too weird to listen to, or you can speed stuff up on videos that you're watching the extra stuff if you just want to get through it. So something to keep in mind. But thank you for that feedback. We'll keep chugging along here. All right, so the next few slides are specific to cell membrane transport. Right? So like I said, the plasma membrane structure itself is one of the more complex compared to the rest of these structures we talk about in this class, because there is so much that goes into it, right? The phospholipid bilayer, all the different types of proteins, some of the carbohydrates that are playing a role, cholesterol, right? We have lots of stuff in this membrane. And so we're going to go into some of the specifics about how specifically like the proteins are used and what type of stuff can just simply cross this membrane. So hopefully you're familiar with the terms diffusion and osmosis. Could I have anyone offer to define diffusion for us? Give an idea of what diffusion may be addressing.
Oh, can you hear me? I can't think of the exact terminology, but it's just where like things naturally like spread out in an aqueous environment, right?
Awesome. Yeah. Yes. All right. So diffusion is describing the natural force that is exerted on stuff when there is space for it to spread out, essentially. Molecules that are dissolved into some aqueous environment, so inside the cell, and most of our extracellular environments are aqueous, water-based. So any molecules in these spaces are naturally going to want to spread out from each other. Okay? And so diffusion is powered by what is called a concentration gradient. And a concentration gradient is anytime there is a difference in concentration within a space or between two spaces that are separated by a barrier. And so we have a barrier, right? The membrane that is surrounding our cells. We have two different spaces, our extracellular space and our intracellular space, so outside and inside the cell. And so anytime there is a difference in concentration, so if there is a lot more of these molecules of these solutes outside of the cell, and there's a lot less of them inside, those molecules will tend to be driven to move towards the space. So they will move from a high concentration to a low concentration. And that natural movement is called diffusion. But there are only a certain set of type, or there are certain characteristics to molecules that are able to just freely diffuse across this membrane. There are lots of other molecules that will not be able to do that. They cannot cross this barrier through diffusion, and thus will need help from a protein to get into or out of the cell. And if they don't get assistance in that way, in order to move with their concentration gradient, osmosis is what is going to be driven to occur instead. And so could anyone expand on what osmosis is referring to compared to diffusion?
I'll give it an attempt. So it's when water follows the concentration gradient of a solvent, right? So if you have a high concentration of a solvent that and you have a selectively permeable barrier, then the water will move towards the area with the higher concentration of solvents compared to water.
Awesome.
Is that correct?
No, that was great. The only thing wrong with it was that you used the term solvent instead of solute. The water is the solvent. Right? So the solvent is what is doing the dissolving, and then the solute is what is dissolved in that space. But everything, the concept you described is exactly that. Right? So when a molecule is under pressure from that concentration gradient, so again, with the example, like if we have a high concentration of something like, let's say sugar, so glucose, which needs help to cross this membrane. We have a high concentration of glucose outside and a low concentration of glucose inside. If there are proteins in the membrane that can help that glucose, then glucose will move. Glucose will move through those proteins. But if it doesn't receive that help, instead, water is going to move. And water moves in the opposite direction. So if there's a high concentration of glucose outside and a low concentration of glucose inside, water is going to move out of the cell. And that essentially creates more space for the glucose to spread out without having to cross that barrier. Okay. But both types of movement are constantly happening at all times. Molecules are diffusing and water is diffusing, so osmosis is occurring at all times. It's just these changes in concentration will drive movements in one direction or the other. So great. Diffusion is that free movement of molecules through some space towards a lower concentration. Osmosis is the diffusion of water, and typically is towards a higher concentration of a solute, so the molecule that is dissolved in that space. And so essentially, molecules that can cross that membrane and thus will simply diffuse based on their concentration gradient are typically molecules that are small and hydrophilic, or I guess if they are hydrophilic, they must be small, is what I'm trying to exemplify here. So if they are carrying a charge, if they are able to bond with, interact with water, they must be very small to be able to cross that plasma membrane through diffusion. So these are really just like ions, so individual elements that are carrying a charge. And then most hydrophobic molecules, so those that do not bond with water, are going to be able to cross this membrane because the inner portion, these fatty acid tails that make up that inner portion of the membrane, are also hydrophobic, and so they can simply pass through. Molecules that are not going to be able to just simply diffuse across the membrane are any large molecules, and especially if they are hydrophilic. So there's a bit of a gray area there where there are hydrophobic molecules that are too large to just simply diffuse across the membrane. But in general, hydrophobic molecules can cross, and then in general, large molecules cannot. All right. And so for these molecules that cannot simply diffuse, in order to cross that barrier, they must be transported by some sort of carrier mechanism, such as a transport protein. Okay. And so here is diffusion exemplified through this image, right? So we're dropping some solutes that is then going to dissolve in this water in our beaker here. So the solute drops in, and then over time, it is naturally going to be driven to spread out until it is evenly concentrated throughout this space. So the goal of diffusion, or what is driving diffusion, is to try to reach equilibrium, which is an equal distribution of solutes within that space. And then osmosis is very similar, except it is the diffusion of water, and again, is moving against that concentration gradient. So it's going to move towards where there is a lot of this solute. So in the example we have here, we have two different sides of this almost ring shape, or this half ring shaped instrument here. We have a selectively permeable barrier separating the two environments. And then we have water and glucose. And so you can see the glucose are these purple dots. So we have a much more of them in this space and much less of them in this space. And thus we have a higher concentration of glucose on side B and a lower concentration of glucose on side A. And so if glucose could cross this membrane, it would simply spread out until we had an even amount, but it cannot. And so instead, water is going to be forced to side B, which is going to create more space on side B and less space on side A. I'm just going to go ahead and skip ahead a slide here to show that. Okay. So if the solute cannot move, water will move, and thus we get more space. And so now that there is less space on side A and more space on side B, the glucose molecules that are on both sides are relatively evenly distributed. And so we have an equal concentration. The only way that osmosis will be prevented or forced against this movement is if force is applied. Okay. So we've applied force now to side B, which is going to squish the water back to where it started. Again, the glucose can't move. And so now we just end up with this concentration gradient that this environment can't do anything about. And we have force applying here, so water can't come over here and take up space. And so these molecules just kind of need to live with this concentration gradient, though they will constantly be driven to try to spread out. Right. And so for most of the systems and processes we'll be looking at, the concentration gradient is either going to drive the diffusion of a small or hydrophobic molecule, or we have some sort of membrane protein that is going to assist with the movement that we need. However, when that is not possible, we do run into some issues due to osmosis. Okay. So again, skipping ahead a little bit. Osmosis can have a detrimental effect on a cell. So when a concentration gradient cannot be evened out by the movement of those molecules, or with the help of a protein, osmosis will occur instead, and that can cause either a cell to shrivel up to the point where there's no space in that cell anymore for it to function, or the cell can swell all the way to the point where it might even burst. So it can have detrimental effects on our cells, and we're going to come back to this briefly. I just wanted to pull up this slide since I was going down that direction. All right. So osmosis is that diffusion of water through a permeable membrane. The term osmolarity is describing the total concentration of solutes in an aqueous solution. So if you're ever given osmolarity, or these terms are much more common in a chemistry course, but the osmolarity of a solution is telling you the concentration of the solute in that solution. And then osmotic pressure, very similar to the pressure from a concentration gradient, The osmotic pressure of a solution is the force with which that water moves into a solution because of the solute concentration. So in this example we have set up here, osmotic pressure is going to force the water towards side B. So it's going to force water to travel through osmosis to side B because of the osmolarity. Right? So the osmolarity of side B is higher than the osmolarity of side A, which creates osmotic pressure, driving water to move from side A into side B. And then the rest of these are kind of just images depicting this idea in different ways. So the main idea here is that if there is a difference in concentration, that molecule, the solute that is dissolved, is naturally going to want to move to where there's more space. And so that is the first thing that is going to be checked. So the molecule will try to move to where there is more space. If the molecule cannot move, the solute cannot move, instead, water will move to create more space for those molecules to spread out. So diffusion of molecules goes towards where there is a lower concentration. The diffusion of water goes towards where there is a higher concentration of that solute. And there's a suggested quick video on osmosis. I believe all the videos that I have linked in this slide deck are also on Canvas, like with those extra helpful videos and links that I posted. So you can find them either way. So back to specifics about the effects of osmosis. This is a particularly prevalent idea to consider when thinking about the blood, right? Especially with like when we are giving IV fluids or intravenous fluids to try to make up for a lack of fluid in a body, right? We need that fluid that we are adding to this new body to be of an equal osmolarity to what is already traveling through our body, through our veins, through our circulatory system. Because we don't want to drive osmosis, we don't want to drive the movement of water into or out of our red blood cells by adding a solution to the circulatory system that is either hypertonic or hypotonic. So having a high concentration would be hypertonic, or having a low concentration of a solute is hypotonic. Instead, we need to make sure that that solution is isotonic, meaning the same concentration. That way, our red blood cells are not losing or gaining excess water. They maintain their shape. The hemoglobin proteins that are carrying oxygen have the room to function and perform their role, right? And thus we have equal movements, or because we have equal movement of water and solutes into and out of the cell. If the solution we are adding to this person's circulatory system is not of an even concentration, so if it's not isotonic, iso meaning the same, it could be either hypotonic, hypo meaning less than, that's a very common prefix that you'll see throughout health and throughout biology, hypo meaning less. So a hypotonic solution would have a lower concentration of solutes compared to the cell. So in this case, there is more of salt in this case, I believe is the solute we're studying. So there is a higher concentration of salt inside the cell than there is outside. And so water is going to flood into the cell to try to make more space, but this could eventually cause the cell to burst or lyse. So this term to lyse means that the cell's membrane has too much pressure from all the water that is stuffed into the cell. The membrane can no longer stay together, so it ruptures and the cell essentially bursts apart. Okay. In the opposite scenario, where the cell is in a hypertonic solution, so hyper meaning more, so there's a higher concentration of solute outside than there is inside the cell. Water is going to be driven out of that cell, which is going to cause the plasma membrane to shrivel together or crenate. This term crenate is referring to this cell shriveling where the plasma membrane all clumps together. And this is going to leave the cell with barely any space inside of it, so no cytosol or very little cytosol. And so very little room for any of the transport or functions that the cell needs to survive. Okay. So both can be deadly to the cell itself. And so IV fluids have a 0.9% solution. of sodium chloride. This is just table salts. Sodium and chlorine are extremely common ions in our bodies and our bloods, or in our blood at this specific concentration. So a 0.9% solution of sodium chloride to water is considered isotonic. That should be the exact same concentration as the blood that is traveling through the circulatory system. All right. Okay. So next we're going to look at the types of transport, right? We discussed diffusion, we discussed osmosis. And so the rest of this is mainly looking at the facilitated versions of transport, right? So we have passive processes, which do not require energy, no cellular energy, meaning ATP. Okay, so adenosine triphosphate, that is the cellular currency or the energy currency within a cell. Any of these passive processes, they do not require ATP. And so that goes for diffusion, that goes for osmosis. These are naturally driven. And then we have our first example of a protein-mediated transport here, which is facilitated diffusion. So this is the movement of a molecule. Sorry, I'm kind of skipping around. So facilitated diffusion, like what we have exhibited here, is the movement of a molecule with their concentration gradient. But typically it is a molecule that could not cross on its own. And so this still requires some energy because we need to move this protein to make it work. But instead of adding energy through ATP, typically some other molecule, some smaller molecule that would naturally diffuse, is going to move through this space as well, which generates thermic energy, generates some heat just from the movement of that molecule itself. And then that energy is immediately used to transport our original molecule. So in this case, we have glucose that we're trying to transport from outside of the cell to inside of the cell. And then sodium, there we go. Sorry, this is the slide I was looking for. And then sodium can act as kind of a helper here, essentially generating energy through movement, going with its concentration gradient to help get that glucose molecule across the membrane. So again, there's no ATP being utilized here as a source of energy. It is simply the movement of another molecule. And I think with this specific example, I'm using that backwards. A lot of the time, the ion is what is acting as a cofactor, helping with the movement. But I believe specifically with our sodium-potassium pump, which is what we have shown here, the goal is to move sodium against its concentration gradient, and it's using the natural movement of glucose to actually power that. So in this case, glucose is acting as a kind of a cofactor to help here. All right. So just backtracking to make sure I didn't skip something important. Right. So that is our last category of passive processes. So that is called facilitated diffusion, because it's still using a protein, but does not require the input of any energy directly from ATP. It uses the movement of other molecules to drive that energy. And then we have examples of active transport or active processes, and these are all the movement that would require energy to move this that molecule across the membrane. Or I guess this is important to talk about. So let me address this first. You are going to be asked to memorize or be aware of the major extracellular ions versus the major intracellular ions, because the movement of these ions is going to play a big role in generating the membrane potential and thus transmitting signals and stimulating the release of molecules like neurotransmitters from out of our neurons. And so the major extracellular ions, so which would be in high concentration outside of the cell, are sodium and chlorine. Sodium has given away an electron to chlorine in its ionic bond, right? So it's a positively charged cation. Chlorine is a negatively charged anion. Those are in high concentrations outside of our cells. Inside of our cells, what are considered the major intracellular ions, in this case, are going to be potassium, phosphate, which is phosphorus with 4 oxygens bonded to it. So this should be a subscript, and I can change that later. And then proteins are typically carrying a charge and playing a role in that membrane potential and the overall electrical charge of our membrane. And so the movement of these ions into or out of the cell plays a big role in what the overall charge of this membrane is. Okay? So the sodium-potassium exchange pump ends up changing our membrane to a certain point, changing the membrane potential to a certain point, at which a large release or movement of ions occurs, causing a really fast change in membrane potential. And that essentially causes an electrical current to move down the membrane. So the pumping of these ions against their concentration gradient causes a significant change in the overall charge of the membrane, which is going to cause a chain reaction, an electrical signal being sent down that membrane. And again, this is used for communication. It's used for the release of other molecules, typically out of one cell, such as the neurotransmitters that are released by our neurons and then used to transmit a signal to the next neuron in some signal transduction pathway. Okay, so active transport. Active transport requires the use of energy. So cellular energy here, our ATP, right, is going to be breaking off that third phosphate group, releasing a significant amount of energy, which allows this protein to then do work, essentially moving the solute against its concentration gradient. So even though there is a higher concentration of the solute inside the cell, we are still trying to move this molecule in. Naturally, the molecule is wanting to be pushed away, and so we use energy to fight that natural force and push the molecule into the cell. Some of the major examples of active transport are what we call endocytosis or exocytosis. Endo is a prefix meaning into. Exo is a prefix meaning out of. And so in this case, into the cell, out of the cell. So these are two different types of movement, bringing molecules into the cell or bringing molecules out of the cell. And specifically, bringing them in through these vesicle structures. So endocytosis is not going to simply move a molecule across a membrane. Instead, it is creating this transport organelle called a vesicle by kind of folding in the membrane, which is what we see the beginning of starting at 1 here. So we have a bunch of these receptor proteins sticking out. They are going to bond with the ligand, so whatever solute we're trying to bring into the cell. And as those ligands bond with the receptors, that is going to start the movement of this membrane as it folds in. So we have it starting to fold in here. It's much more folded now until eventually the cell membrane here is going to connect again, allowing this completely surrounded membranous vesicle to pop off or pinch off into the cell, right? So we have not brought anything across this membrane yet, right? They are still in their own environment, separate from the cytosol or cytoplasm, because we have completely surrounded it with our plasma membrane and then pinched off to bring that vesicle inside. And so a lot of the time, this is what is used for like cellular digestion. So this vesicle is going to then fuse with another very similar structure called a lysosome. So this lysosome is essentially a vesicle that is full of digestive enzymes, proteins that are going to break apart whatever this solute is. And then we can release those smaller bits and pieces into the cell itself, or we can recycle them by sending them back out of the cell. So for instance, there are immune system cells that need to kill invaders like bacteria and viruses, do a form of endocytosis that we refer to as phagocytosis. So it is engulfing this bacterium with a plasma membrane, bonding with a lysosome, which is going to release those digestive enzymes. Those enzymes kill the bacterium, and then we can release all of the proteins and leftover junk from that bacteria back out of the cell, right? Thus reducing the probability that a bacterial infection occurs. And so essentially the same concept is true in the reverse for exocytosis. If we have a bunch of molecules that we want to remove from the cell without releasing them into the cytoplasm, we can carry them in a vesicle. It will bond its plasma membrane with our own cell membrane, or sorry, it will bond with its organelle membrane with our plasma membrane. And then that will allow for the release of those solutes back into the environment without them ever mixing in or traveling through the cytoplasm. They're always surrounded by a membrane. So here's a microscope image of that happening. So you can see that kind of pinching occurring of our membrane here, where stuff can flow in here, but eventually those two membranes are going to connect, and this will connect, and thus this vesicle gets to pinch off like this. So now some quick specifics about some of the other major organelles in the cell. So we just have a couple slides on the endoplasmic reticulum, and then a few slides on the Golgi apparatus. So the endoplasmic reticulum, we have a couple different types that are both referred to as the same organelle. They have the same general functions with some slight differences. So in general, the endoplasmic reticulum is responsible for the synthesis and modification of, so creation and adjustment of proteins, lipids, and carbohydrates. And so mainly this is the synthesis of all these things, and then the modification of mainly lipids and carbs. Protein modification is mainly occurring in the Golgi apparatus. Okay. But so the endoplasmic reticulum is really important for creating larger versions of those biomolecules that we introduced. Right? So creating our large polymers of proteins, lipids, and carbohydrates. And so for the protein portion of that, that is mainly happening within the rough ER, or the rough endoplasmic reticulum. The rough ER is given that name because its structure under a microscope looks rigid and rough, like what we see here. Because it has a bunch of ribosomes attached to it. They are directly embedded into the membrane of this rough endoplasmic reticulum. And ribosomes, as we mentioned briefly earlier, are the sites of protein synthesis. They make proteins from the instructions within our genes. And so the proteins made on the rough ER can be sent directly into the internal space of the endoplasmic reticulum, where they can be further modified or packaged up and then sent out to where they need to go. The smooth endoplasmic reticulum does not have those ribosomes, and so is mainly going to be working with and adjusting lipids and carbohydrates. And so we can see that here. Right? So our rough ER with all these little bumpies from the ribosomes, and then the smooth ER, which is much smoother since we don't have ribosomes on the membrane. A major portion of the endoplasmic reticulum is directly attached to the nuclear envelope, so our membrane surrounding the nucleus here. And that makes it really efficient for that protein production. Okay, so the genes, the instructions that are protected within our nucleus can be transcribed into mRNA. That mRNA can go directly into the endoplasmic reticulum and bond with a ribosome to start creating the protein that gene coded for. But we do also have portions of the endoplasmic reticulum that are floating throughout the cytoplasm, separate from the nuclear envelope. Right. The Golgi apparatus looks really similar at first glance. We have a bunch of these folds together, like we did with our rough ER and smooth ER, a bunch of these folds and spaces. But the Golgi apparatus is always separate from the nuclear envelope and acts essentially as the postal system of the cell. So the Golgi apparatus is going to be creating vesicles, right? Those membrane-bound carriers we talked about a little bit earlier. It's going to be creating vesicles and attaching a signal to it, whether it's a carbohydrate or a protein, that is going to direct where that vesicle is going to end up. So whenever we are producing new molecules, they will be transported to the Golgi apparatus. The Golgi apparatus will create a vesicle with some signal on it, and then that molecule will help it, or that vesicle will help the molecule get to where it's needed. And so same thing for stuff that's coming in. If we have now endocytosed some molecules like glucose that we need to get closer to the mitochondria so they can go through glycolysis and then be transported into the mitochondria. For the rest of cellular respiration, they will first go through the Golgi apparatus, packaged into a vesicle with a signal, and then sent towards the mitochondria. But a second function of the Golgi apparatus is that it is the main organelle that is going to be responsible for protein modification. So in order for our proteins to finalize into that tertiary or quaternary structure where they are now functional, that is typically happening within the Golgi apparatus. Here's another look at that here. So this next series of a few slides is just a broken apart figure from the text, kind of going through the endomembrane transport system. So showing the series of stages or the steps that a protein would go through as it goes from being translated and then through the Golgi apparatus, et cetera. So how things move throughout the cell, essentially. So protein synthesis has to start with the protein itself actually being produced. So we're going to have a few slides talking about transcription, which is the copying of DNA into mRNA. That mRNA leaves the nucleus to go to a ribosome, and then a ribosome will be able to translate those instructions. So the second stage is called translation. It translates that blueprint, that mRNA, into a series of amino acids. So by the time a protein is done with the ribosome, we just have this long string of amino acids, that primary protein structure, which is still what's being shown here. This may happen at the rough ER instead. Okay, so that primary structure, so this chain of amino acids, which you could refer to now as a polypeptide, is either going to be released into the rough ER if it was a membrane-bound ribosome here, or just released into the cytosol. Okay. They'll start to fold into its secondary structure, or be brought into the rough ER for help with folding into that secondary structure. And then the endoplasmic reticulum is going to package it into a vesicle and send it to the Golgi apparatus. So we now have a protein in its primary or secondary structure, so not yet functional. It was packaged up by the endoplasmic reticulum, sent out into the cytosol to travel to the Golgi apparatus. And then within the Golgi apparatus, it will be modified and so shaped even further into its tertiary or quaternary structure, where it is now a mature or functional protein. The Golgi apparatus will then make another vesicle to carry those proteins with some sort of signal sticking out of the vesicle itself, and that will help bring that protein to where it's going. So in this case, it looks like it's showing us the production of a lysosome. So these proteins have been activated as digestive enzymes. They are packaged up into a membrane-bound vesicle here. And so now this lysosome is available for whenever we need to do cellular digestion. So break down some molecule, like what we were talking about up here. Okay. So we just produced this lysosome. We have a vesicle full of digestive enzymes that can help us break apart any molecules that were sent in. I'm getting a phone call. Sorry, I don't think I've ever gotten a phone call while I've actually been in the office. Okay. Give me a second, guys. Okay. Apologize for that short interruption. Okay. So that is essentially the general movement of proteins from production and through that endomembrane transport system. So produced at the ribosome, packaged up in the ER, sent to the Golgi apparatus for further maturation, and then sent back out from the Golgi to wherever they are needed. Great. So moving on to the mitochondria. So the mitochondria is an organelle present in all eukaryotic cells, and especially important for all of them, but especially important in animals since we don't have any other direct energy producing organelle. And the mitochondria is going to be responsible for producing a majority of our ATP, our cellular energy. A mitochondria is not needed for the production of ATP. There are cellular processes that can produce ATP within the cytosol, or in some of our other organelles, but the mitochondria is especially efficient at it, and produces much, much more than any of the other ways of producing ATP in the cell. So the mitochondria is responsible for metabolizing substrates of lipids, proteins, and mainly carbohydrates. And so specific here with the word substrates, meaning lipids even in their monomer form, so a glycerol and a fatty acid, those two things cannot enter the mitochondria yet. They would need to be transformed into another substrate, so further broken down a bit into either acetyl-CoA or pyruvate. And the same thing with proteins. The certain amino acids can be transformed into acetyl-CoA. Other amino acids can be transformed into pyruvates, okay? But they must be changed into those forms before they can enter the mitochondria. And so the same thing goes for carbs too. So carbohydrates are the main source of energy throughout a cell, especially during activity. But even a carbohydrate like glucose needs to first go through glycolysis to be split in half into two pyruvate molecules before it can enter the mitochondria. But the mitochondria is essentially specialized to do just that, to break down those substrates even further and create a whole bunch of energy as a result. And so the mitochondria is unique to every other organelle in animal cells because it is the only one that has two membranes. So we have an outer membrane and an inner membrane. And I'm not referring to the phospholipid bilayer. So all membranes in our cells have two layers of phospholipids. So I'm not just talking about two layers. This outer membrane has a bilayer, this inner membrane has a bilayer. So these are two separate membranes. And so this creates even further compartmentalization or specialization of the space within this organelle, within our mitochondria. So we have our intermembrane space, which is all the space in between the inner and outer membrane. And then we have the matrix, which is the term for the space in the very center, so inside of our inner membrane. And so again, this allows for a lot of control. So movement of molecules across this inner membrane is only going to affect the concentration inside the mitochondria. And that's ultimately going to be really important for the very last stage of cellular respiration, which is what produces a majority of our ATP. So cellular respiration in a nutshell is composed of three stages. So glycolysis is what happens outside of the mitochondria. So the very first stage is starting with a glucose molecule and splitting it in half. And as a result of splitting it in half, so that one glucose, the carbons that are in that glucose become two different pyruvate molecules. But since we have broken the bonds, the non-polar bonds holding those carbons together, we are releasing the potential energy that was stored in those bonds. So anytime you are breaking carbon-to-carbon bonds, you are releasing potential energy. Or generally, if you're breaking bonds at all, you are releasing potential energy. But as far as the transformation of energy in organic molecules, that's almost always going to be carbon-to-carbon, at least for respiration here. So we split glucose in half. That released a little bit of energy. Some of that energy is going to be immediately used to make ATP. So glycolysis on its own, without the mitochondria, can make 4 ATP. However, it requires 2 ATP to begin with. So really, we only get 2 new energy molecules from glycolysis. So not a whole lot, and very inefficient. And if these pyruvate molecules then cannot continue through cellular respiration, they ultimately end up using even more energy to convert them back to glucose in the liver. So glycolysis on its own is very inefficient, but we do get a little bit of energy from it. So once glycolysis is finished, we have our 2 pyruvate molecules, which are going to continue into the mitochondria if we have oxygen. So essentially the whole reason that you breathe in oxygen and then breathe out carbon dioxide is for this process of cellular respiration. Oxygen is needed for the final stage of cellular respiration, and thus pyruvate will only be brought into the mitochondria if oxygen is present. And then as a product of breaking down these molecules to get ATP, all the carbons that we started with are going to be released back out into the environment as carbon dioxide. So you breathe in oxygen to allow for this process to occur. You breathe out carbon dioxide as a result of breaking down these molecules. Right. So we said glycolysis happens outside of the mitochondria. It only gives us a little bit of ATP, and our glucose was split in half into 2 pyruvates. The other important information here, or the other important piece of this equation, is what we call electron carriers, electron carrier molecules, like NADH. NADH is essentially holding on to a hydrogen that was from that glucose molecule, and is going to carry that to our final stage to help us produce a whole bunch of energy. And so we have NADH produced during glycolysis, and then we have NADH and FADH2. Sorry, I thought I heard something. I do hear something. Do you guys hear that slight? Is something playing in the background for you guys? Sorry, that's gonna drive me crazy. Where is that coming from? Aha, okay, sorry. It was my watch started playing YouTube. I lost my train of thought. We were talking about electron carriers. So these electron carriers are essentially carrying hydrogens that were originally present in whatever molecule we're breaking apart. So in glycolysis, NAD plus picked up some electrons, some hydrogens from that glucose. That is going to further occur even more during the citric acid cycle, which is the second stage here in the matrix of the mitochondria. We produce even more NADH, and then this other electron carrier, FADH2. And again, the whole point of these is to carry those high-powered electrons from those hydrogens. These are essentially hydrogen ions, carrying those high-powered electrons to our final stage of respiration. So pyruvate came all the way into the mitochondria, all the way across the outer membrane, across the inner membrane, and into the matrix. It gets broken down into acetyl-CoA, and then acetyl-CoA goes through the citric acid cycle. And so eventually, through the citric acid cycle, all of the carbons that were originally in that glucose molecule have now been broken down to carbon dioxide. And as a result, we have a whole bunch of these electron carriers now waiting for us. We've only made a little bit of ATP through glycolysis, and now we just have a whole bunch of these electron carriers. And so the final stage of cellular respiration is where we get to use those electron carriers, the electrons from those electron carriers, to make a whole bunch of ATP. So essentially, the electron carrier molecules are going to donate their electrons to a series of proteins along this inner membrane called the electron transport chain. And so the electrons move from protein to protein down this chain, thus the name. We're transporting electrons across a chain. They will move from one protein to the next until eventually they end up on oxygen. So if you remember from us talking about covalent bonds, oxygen is super electronegative. It's very needy and greedy with electrons. And so oxygen is really driving the movement of the electrons from one protein to the next. And so ultimately it will take those electrons and become a water molecule. So we produce water as a waste product here through this electron transport chain. But as far as what's important for energy, every time the electron moves from one protein to the next, a proton, so this H plus, this hydrogen cation, this proton is going to move from the matrix into this intermembrane space. And so by the end of all of our electron carriers donating their electrons and those electrons moving down the chain and making water, we have pumped a whole bunch of protons into this intermembrane space, leaving us with a really high concentration of those protons. And so these protons are going to want to move back to where they started in the matrix, where there is a low concentration. So we've created a concentration gradient. And the only way the protons can move back to where they started is through a special protein called ATP synthase. And the movement of those protons generates thermic energy, which drives the movement of this portion of ATP synthase. And that rotation creates ATP from ADP and our free phosphates. So the protons are acting as fuel flowing through this ATP synthase engine, causing this rotor essentially to spin and generate a whole bunch of ATP. Right? And so from the electron transport chain, we generate like 30 to 40 ATP molecules. It's hard to give an exact number from this entire process because energy gets lost every time a chemical reaction is occurring. And sometimes these reactions are occurring more efficiently than others. So we do lose energy through this process. The total amount of ATP produced varies. But in general, the electron transport chain is making about 34 ATP molecules, or ATP synthase specifically is making about 34 ATP, and glycolysis only made about 2. So when oxygen is present in the cell, and we can use the mitochondria to do the citric acid cycle and the electron transport chain, our cells are producing energy, ATP, much more efficiently than they are without oxygen. So oxygen is super, super important. These next couple slides just have bullets essentially explaining what I was just talking about on that slide, right? Our electron carriers donate those electrons to our proteins along our chain here. Electrons are passed from one protein to the next until they ultimately end up on oxygen and become water. Every time an electron moves from one protein to the next, a proton is pumped into the intermembrane space, thus creating that concentration gradient where we have high concentration of protons here and a low concentration of protons in the matrix. That concentration gradient drives the protons through that channel, ATP synthase, generating thermic energy and allowing for ATP synthase to make new ATP. So we take an individual phosphate group and add it onto an ADP, which already has 2 phosphate groups, and now we have ATP with 3 phosphate groups. And that ATP can travel throughout the cell to wherever energy is needed, break the bond holding those phosphates together, and release the energy for that structure to then do work wherever that is. This is just another look at that protein. And so I mentioned earlier that the movement of kinesin, that transport protein along microtubules, was one of the first things that got me excited about biology. This protein, ATP synthase, is the thing that convinced me I need to switch majors or add a biology major onto my psychology major. Just the idea that this super critical, very, very prevalent and common protein that exists in all animal cells, all plant cells, all eukaryotic organisms, that this protein acts exactly like an engine that we use like in our cars, or I guess a basic version of the engine that we use in our cars, blew my mind at the time as a 18-year-old college student or 19-year-old college student, however old I was when I learned it. But literally protons acting as fuel, causing the rotation of a rotor, generating heat and energy to then do work like drive a car, was really cool to me. So if you're interested, check out some videos on ATP synthase too. In general, for biology content, I highly recommend, or basic biology content, like what we're going over here. I highly recommend Amoeba Sisters or Crash Course Biology as YouTube channels that both make really great animations in their videos and are very entertaining as educational videos. So I highly recommend you check those out. And that's the last slide we'll go over for today, since we only have a couple minutes. I'm not going to jump into the nucleus. So we got through 69 slides out of 99. So we only have 30 more to go. So I will put together a short activity highlighting some of the more complicated processes that we talked about in here. So with that being said, does anyone have any questions or comments that we can address in these last couple minutes here?
How much detail are we required to know about the Krebs cycle?
About the Krebs cycle itself?
Yep.
Not much. So knowing that it takes an acetyl CoA molecule, so acetyl coenzyme A, and then pumps out electron carriers, and all the carbons have now been removed as carbon dioxide. That is the extent of it, right? So I know there's other figures you could pull up about the citric acid cycle that have every single chemical reaction that's happening in this long chain of chemical reactions. I'm not requiring you to memorize all of that. No. So just this part really. Any other questions like that too? Those are important for everyone, right? And understand what is expected. So as you guys have probably noticed with the first quiz, like the chemistry quiz, the quiz questions and answers are extremely specific, like they are rigorous quizzes. Because similar to how the idea of lab is to get you into the practice of analyzing, observing in detail, and then reporting in detail, it's kind of reflecting that same practice here with the lecture content. Right? The quizzes are really tough and very specific. And so you get unlimited attempts to ensure that you are spending a good amount of time thinking about that detail, the very minute specifics. On an exam, the questions aren't necessarily going to be at that critical of a detail, but hopefully through the practice of answering questions in that detail, it helps develop the actual understanding about that idea, which will translate to the exam. And hopefully that makes sense. Right? The exam questions won't be as critically detailed. There's a lot more open-endedness in my assessments, giving the opportunity to show understanding in different ways rather than just factual reproduction of information. So I hope that provides a little bit of clarity there. I got to look through the first set of quiz responses and saw that a large majority of this class are at a 3 or below in their confidence with chemistry. I think there are only 2 students that rated themselves above that. So please also take advantage of office hours if you have questions. And again, recognizing that the level of detail on the quizzes is not exactly what you are expected to retain for the exam, for the assessment. Yeah, the other thing I wanted to make sure to note was make sure you do the peer reviews for the introductions video assignment. So if you submitted your recorded video to that discussion board before the due date, you should have been automatically assigned 2 peer reviews through Canvas. I've included a YouTube video in the assignment instructions that goes over, from a student's perspective, how to access those peer reviews. But make sure you are aware that your peer review is due next Sunday. Okay, so just going in and commenting on both of your assigned peer reviews, showing that you have watched their videos. Yes, Ben.
Yeah, I'm not sure you'll know the answer to this question, but I watched the video, and in the video they said that you had to find it through your Canvas email, the email that's attached to Canvas. Is that the only way I can access my, that assignment, the peer review stuff?
It shouldn't be. There's also written instructions. Let me pull up. I believe I included written instructions in there as well. But you should be able to do it through the discussions tab. And if anyone else has been able to do that and would like to share how they did it, since you have more
I was
recent.
gonna bring that up. I didn't see who I was assigned to, so I don't know if I'm not saying something or.
Okay. Then in lab, 'cause it's hard for, like, I'm seeing the instructor view, and even when I go into student view, like the fake student didn't upload a video, so it wasn't assigned peer reviews. So it's hard for me to see from where I am exactly what buttons you guys are seeing. So in lab tomorrow, I'll maybe with you, Ben, I'll have someone go through their Canvas with me, and we can work together to find where that's located. And I'll try to share with everyone in lab right then and there. So thank you for notifying me of that it wasn't coming to you through your email, and you're having trouble finding it.
I'm just seeing mine on my there's like a to-do list on the right-hand side.
Mhm.
I'm actually seeing it on that drop down, kind of on the main biology page. I say
And when you
peer
click
review.
that, it takes you to the peer reviews, or it takes you to the same discussion. You're muted. You're muted, James.
Sorry about that. Yeah, no, it actually does. If I click on it, it takes me directly to the peer review person that I'm supposed to review.
Okay, cool. So try that out through the to-do list on the homepage when you come into Canvas, or when you come into our Canvas site. But we can still explore that in lab tomorrow too, if anyone is still struggling with it. But that's all of our time. So unless you have any other burning questions or comments, you guys are free to go. I will see you tomorrow to talk about the bones of the skull and face.
Thank you.
Have a good one.
Thank you, sir.